Okay, welcome to Bio106 Spring 2021 semester here at Hagerstown Community College. I'm Dr. Murphy. I'm an instructor. You may, if you're going here at the week of January 11th, you may not have seen me yet. We're going to have some Zoom sessions in the first week of the semester, which is postponed to next week. But I want to make some of these video lectures available to you even before we officially start the semester, just to maybe get you introduced and get maybe get you a head start on the semester. So these are going to be the lectures. We're going to have, well, the first 4 weeks we're going to have even the labs are going to be online. Hopefully, if the transmissibility rates in Washington County go down enough, we'll start having on-campus labs in about 4 weeks, perhaps. Let's hope. But we got to deal with what we have right now. So, but all the lectures are going to be video. I'm going to make lectures for each chapter from this book right here. Here's our textbook, Sensory Biology, Campbell, 7th edition. And I have PowerPoints from the publisher, and I made them available in D2L, and I'm going to go along here and discuss the chapter 1. And these video lectures, like I said, there'll be several for each chapter, usually only be about 30 to 40 minutes long, and I'll have multiple ones for each chapter, and then they will be put up in our YouTube site, my YouTube site, and that URL is in the syllabus. So then you can go to YouTube site and download them and listen to these lectures whenever you want. Okay. I'm going to do something now called a screen share. I am going to pull up the PowerPoint for chapter 1 and share my screen with you. And so my picture will be up small in the corner, and most of the screen will be the PowerPoint presentation. So let me do that right now by sharing my screen right here. So there's the PowerPoint, and I'm going to from beginning, and there it is. Learning about life. So we could start with scientific study of life, an overview and a process of science. This first chapter is really an introduction to science. You know, what is science? How does science work? All right. And biology is a science, the scientific study of life. And what does it mean to think about something scientifically, or how do you tell the difference between science and other ways of knowing about the world? Science is just a way of knowing, it's a process. I think the next slide shows it, talks about it being a process, an approach to understanding the natural world. To try to figure out, actually, what's real and what isn't in the natural world. Sometimes it's obvious what's real and what isn't. A lot of times it isn't. It's really, you have to investigate them, collect data, and do experiments and observations to try to figure out, as I said, what's truth and what isn't. So it begins with exploration, and you explore something and you make an observation of some natural phenomena, and you're interested. We're really curious species. We want to know how things work, and to figure out how things work has changed the human experience. It's helped us in incredible ways, the scientific process. Okay, so recorded observations are called data. Also, the information required from experiments is called data. And most people think you have to do experiments to do science. You don't necessarily have to do experiments. You can make detailed observations, you can make predictions, and you can record things. Okay. Launching point of the next step in the process of science testing. Okay, so here's Jane Goodall looking at her chimps. Sometimes, as I said, in science, you can't do experiments. You can't, you just have to watch and make careful observations and write down everything and make predictions and see if those predictions come true. So it's a process. It's a process. Making observations, asking questions, seeking information. So hypothesis. So the process, how you conduct science, a scientific method. First, you make the observation, then you try to think of a possible explanation for that process. You know how things work in the world. Maybe this is what's happening. So this isn't like an educated guess, a hypothesis. It must be testable and falsifiable. That is, you should be able to test it somehow, either with experiments or with observations or with predictions. And then it has to be falsifiable. You have to be able to disprove it. We're going to learn when we get to science that you really can't absolutely prove anything. You can only with 100% certainty. It's all, but you can disprove things with 100% certainty. And in order to investigate something scientifically, you have to be able to disprove it. Okay, some hypotheses lend themselves to experiments or scientific tests. Other hypotheses can be tested by making further observations, and the results of experiment observations will either support or not support the hypothesis. You collect data, you look at what the data tells you, and then you either accept the hypothesis, saying, okay, I think I guessed right, or the data shows something else is going on. We reject the hypothesis, and we come up with a new hypothesis. Something else is going on. So testing a common problem using the process of science. This is a trivial example of how we actually use the scientific process in everyday life. We just don't think about it. It's just like organized common sense. Exploration. You try to turn on a remote. The hypothesis is, oh, the batteries must be dead. Prediction. If I replace the batteries, the remote will work. Experiment. I replace the batteries with a new one. The remote works. Accept the hypothesis. That's what the problem is. However, the remote still doesn't work. There must be something else wrong with it. I reject the hypothesis. So forming a hypothesis, making predictions, running experiments. The experiment was put the new batteries in and see if it works. Interpreting the data, drawing conclusions. All right. So here they jump into scientific literature in fine-tuning experiments. Science requires open lines of communication. Everybody talks to one another, and everybody's clear and open with everything. And that helps everybody come to a collective conclusion or theory about how something happens. What they do is, after doing an experiment, scientists publish the results in a scientific journal. The scientific journal goes out to what are called referees. This is called peer review. Out to experts in the field, and they try to criticize it. They try to find something wrong with it. And they go back to the editor with one of three things. So this is good research, you should publish it. It's lousy research, it's terrible, you shouldn't publish it. Or it has good aspects and bad aspects. If you send it back to their authors and they fix the problems, then you should publish it. Okay, communication, sharing data, taking feedback, publishing papers, replicating findings, building consensus. So you have to publish a paper and tell people what you did, how you did it, and if the data is real and accurate, somebody else should be able to take the same materials you had, do the same experiments, and come up with a similar outcome. That is, so they should be able to be replicated. They can't be replicated, something's wrong. And you build a consensus. You get people start to look at your data, they replicate it, they find out, oh, this seems to work. We form a theory about this, and this is building consensus. People start to agree with you and say, okay, we all agree that this is how this works, how this natural phenomenon works. And that's really a scientific theory. It's a consensus of all the people who've done work in that field and they know something about it, and we all collectively say, okay, we think this is truth. But it's always tentative. Remember, you have to be able to change your mind. Some of the best theories can be, they have to be able to be disproved, and they can be disproved with the right experiment. If they are disproved, then you got to go on to a different theory. Okay. Outcomes, building knowledge, solving problems, developing technologies, benefiting society. I mean, science, how much has science benefited society? Just to look in the human condition, if it wasn't for modern surgical technique, I would be both blind and crippled right now. I am looking at this camera right now through little plastic lenses that have been put into my eyes. I had cataracts in both eyes, and now I have these little plastic lenses that gives me, well, gives me pretty good vision. I still need glasses to read things, but other than that, my vision is great. I have an artificial hip, lost through arthritis. And so, and I never even think about it. It's like works perfectly. And this is science at work. Even, using this technology here to do these video lectures with your cell phones. This is all scientific investigation that have come up with all these fantastic devices, fantastic aids to human society. Okay, overview of the process of science. The scientific method: exploration, making observations, asking questions, seeking information. Testing, forming a hypothesis, making predictions, running an experiment if you can, gathering the data from the experiment, interpreting the data, drawing a conclusion to either accept or reject the hypothesis. Building knowledge, solving problems, developing new technologies benefiting society, and then sharing information. Publishing papers, building a consensus. And unfortunately, nowadays, there are some really scientific experiments and scientific information. I'm talking too fast. Sometimes I tend to talk too fast. I got to be careful not to do that. Let me know if I start talking too fast. You can always email me and say, okay, I watched your lecture and you were talking a bit too fast. I'm not sure what you were actually saying. I can redo something. Some scientific information now is can make a lot of money if you can actually modify genes and come up with certain technologies to make money. So then you don't want to share that up with other people, and that goes against science, really. But mostly it's a small part. Okay. So let's get down to theory. I've already mentioned a theory. Now, in the popular vernacular, theory is, many people use the term theory as a hypothesis. Oh, it's a guess. It's just a theory. But in science, theories are very strong. They're comprehensive, well-substantiated explanation built upon lots and lots of evidence. Science is evidence-based. Give me the evidence. You say this is what's happening, show me the evidence, all right? And I'll make my own decision. So theory is the consensus, as I mentioned. It's what most of the people in the field think is real. All right. Become widely accepted by scientists only if they're supported by a large, varying, growing body of evidence. But theory can be refined and even rejected if somebody else comes up with a better explanation that fits the evidence more. I'll give you a good example of that. The theory of gravitational attraction by Isaac Newton. He's talked about how if I hold this book up and I let it go, it's going to fall. That's a fact. Why does it fall? Well, that's what Isaac Newton was trying to figure out. And he came up with a really good theory about it, about the theory of gravitational attraction. And he figured out, well, if I can remember my physics that I took, once I drop this from a certain height, I can figure out how long it's going to take to hit the floor, how much force it will generate when it hits the floor, how fast it'll be going when it hits the floor, and so on and so forth. And that lasted for centuries, because he came up with it, I think, in like the 15th or 16th, maybe 17th century. But then a guy named Al came along in 1905 and said, okay, Newton was wrong. Well, it was Al Einstein who came up with the theory of relativity that explains why that drops. It's not through gravity. It's because gravity doesn't pull it down. Gravity warps space-time. And so that's why that falls. And I'm not even sure what that means, what space-time is, but he completely revolutionized the whole idea of why something falls. And so even though you had this great theory that explains things so well, that lasted for centuries, one guy comes along and we changed our mind. And you gotta be able to change your mind. A lot of people in science think that's a weakness. Oh, they're changing your mind. Of course you gotta change your mind. You gotta be open. You gotta be open to new ideas and new ways of looking at things and new data. So even the best theory can be disproved. And Einstein disproved Newton's theory. And it has been tested many times, Einstein's theories, and every single time it's passed. So now it's what's the consensus as that's how gravity works. Okay. A fact. Piece of evidence considered to be objectively true based on current evidence. So like I said, I hold up the book, I release it, it's going to fall. That's a fact. It's distinct from opinion. It isn't my opinion if this book's going to fall. It's a fact. Right. Many people associate facts with science, but accumulating facts is not the role of science. Using facts to help plan experiments or trying to provide evidence for a particular theory, that's what facts are for. Okay. Cornerstones of science are the explanations that apply to the greatest variety of phenomena. Okay, to investigate a hypothesis, researchers often test multiple times with variables. A variable is something that, well, can change. And we used to think back in, hundreds, even thousands of years ago, that things really didn't change that much. It was a pretty stable world. It was like a clockwork universe, they used to call it. Now we know that everything changes. Everything is a variable. And what you look at in experiment is you look how variables relate to each other. Variables are factors that change in an experiment. Controlled experiment is one that comprises two or more groups that differ from only one variable. So here's the thing. Science is a comparative thing, or I should say experiment. Scientific experiments are always comparable. You have to compare two groups, one that gets the treatment and one does not. Like I said, you can always see, you can say, oh, I have a drug that will cure eczema. So we just get a bunch of people with eczema and put it on and see whether the eczema gets better. No, you can't do that. If you want to test whether it really affects this, you have to get two groups of people that have eczema. And one group, the control group, gets a cream that looks exactly like the real stuff, but it's not. It's just, it has none of the medicine on it. Versus the other group, the experimental group, that gets the actual medicine. And then after a while, you compare what the eczema looks like in the two groups. So the control group actually does not get the specific factor. An experimental group has or receives a specific factor. And both groups have to be exactly, well, you gotta try to minimize variability between the groups. When I have everything the same between these two groups, the control group and experimental, except for the one thing that you are investigating. Right? That's what you want to be the only difference between these two groups. Can you do that? Absolutely. No, you can't control for everything, but you try to control for as many things as possible. Okay, do baby turtles swim? So, after emerging for 2-month incubation, hatching go out of the sand and use moonlight to navigate the sea. Do they swim in the ocean currents or do they just passively drift? So, attach tiny satellite tractors to 24 sea turtles. Turtles move faster and along different routes than drifting buckets, so as they travel by swimming. So you have we go to the next one. So you tag baby turtles, and then you tag drifting buckets. You put them out in the same place, and just watch them and track them as they go. Do they travel the same distance? So a little baby swimming, or are they just drifting? And looking at the graph to the right here, you'll see that the red line average of 24 green turtles, average of 24 drifting buckets. The turtles are going much farther than the drifting buckets, so therefore they must be actively swimming. So you have to, like I said, it's comparative. So you can think of the buckets as the control group, and the turtles as the experimental group. And you put them out in the same body of water at the same time, and track them that way. So that was a controlled experiment. And so scientific experiments have to be controlled. The independent variable is what is being manipulated by the researchers. In this case, the object under investigation, either turtles or buckets. The dependent variable is the response, output, or effect on the investigation. In this case, the speed of movement. Are the turtles traveling faster than just drifting buckets? And so you only test one independent variable at a time. And usually the independent variable, on a graph, the independent variable is on the x-axis, the dependent variable is on the y-axis, as you can see in the previous slide. The y-axis talks about how far the turtles drifted. Okay. Many medical drug trials include a placebo, a medically ineffective treatment that allows the placebo group to serve as a control group. So everybody knows what a placebo is. It's and the placebo effect is a fantastic thing. It really is. I always say, if let's say you had a splitting headache, and I was a trusted physician, and you really trusted me. And I said, I got this drug here that's going to knock that headache out. I mean, completely. But actually, all this is a sugar pill. It doesn't have any drug in it. But you buy into that. You take that pill, and 9 times out of 10, that headache will be gone because you bought into it. Right? So it's a great thing, the placebo effect. But you got to be careful about the placebo effect in scientific experiments. Unless you're testing the placebo effect, the placebo effect will give you false positives. So you got to make sure that what they do what's called a blind study, and it's even better, a double-blind study. In other words, particularly if it's humans we're talking about. So you have 2 groups of humans. One of it's in the control group that's not going to get the medicine. The other group is in the experimental group that's going to get the medicine. You can't let them know what group they're in, right? Because if they're in a control group, they can say, "Oh, I'm not getting it. I'm not going to get any resolution to this problem." If they were in a experimental, they say, "Oh, I'm getting this medicine. I'm going to get better." So you can't let them do that. And then, so that's a blind study. But then also, the people who hand out the pills to people, they can't know either. So they're blind. They're just handing out these pills to certain subjects. And somebody in the back room made up these pills and designated, participant 1 over here gets this one, participant 2 gets this one over here. And the people handing out, they don't know either. That's called double blind. And that is the gold standard for doing a medical trial. Like testing these COVID-19 vaccinations. They're doing double blind studies. So, type of study, not blind, test subjects know which group is which, researchers know which. Single blind, test subjects don't know, the researchers do know. Double blind, neither one knows. And that's, like I said, the gold standard. That's what you really want to use. Okay, I'm going to take a break here and get on to pseudoscience in the next video. So I'm going to stop right now. This is the I guess we'll call it science number 1, and then I'm going to stop sharing and stop recording, and we'll pick this up with this slide in the next video. I'll see you later.